So today what we're going to first look at is the evolution of the eukaryotic cell. And that's going to be the main idea of this image here. First thing that we want to look at are the types of cells that originated. Evidence shows that our first cell type was actually prokaryotic and not eukaryotic. And this should make sense because our prokaryotic cells are much more simple than our complex eukaryotic cells. And so, as it turns out, our earliest or most primitive cells actually had no internal membranes. So when you think about how we compared our prokaryotic cell to our eukaryotic cell, remember that our prokaryotic cell had no organelles, had no membrane-bound structures inside of itself. And so that's just what we are looking at here. But as it turns out, as our cells start to develop and change, there actually was a trend that led to the increase of membrane surface area, or we saw a trend of increased membrane surface area, SA, and of the internal membrane infolding. What this means is that as our cells became more complex, some of the actual cell membrane or plasma membrane would actually fold inward, creating more surface area within the cell. We're going to talk a lot later on about how that increase in surface area allowed for an increase of work or of chemical reactions to occur. So the more space you had, the more work that the cell could do. Now, when we're looking at the idea of the evolution of the cell from prokaryote to eukaryote, we actually have a theory called the endomembrane theory. This theory says that initially we had many different types of prokaryotic cells, bacteria, that actually cohabitated. So they all lived together. And from those cells, we actually had some oxygen-breathing bacteria that represented our aerobic cells, okay? With the idea that those aerobic cells would need the oxygen and be able to turn that into energy through respiration. And we had photosynthetic bacteria that represented our autotrophic cells. So our autotrophic cells go through photosynthesis, produce oxygen, and then these oxygen-breathing bacteria were able to evolve, or were able to evolve into air-breathing bacteria. And eventually what happened is that these two bacteria somehow were able to move inside of this cell. And so when you're actually looking at how these membranes were able to fold in, this is actually called invagination, the folding inward of the membrane. And so with these two, it's almost like the endocytosis. They were taken into the cell. And for some reason, our cell didn't have any lysosomes or digestive enzymes that could break down that aerobic and autotrophic cell. So then we ended up developing what eventually became our mitochondria and chloroplasts. And so we do have some evidence that supports evidence that cells change over time. Okay? And our evidence is actually, first of all, that our mitochondria and chloroplasts actually have their own DNA. And they can actually replicate their own DNA. The other thing is that the mitochondria and the chloroplasts also have their own double membrane. And because they have their own DNA, they're actually able to also have their own proteins. So here's some evidence that supports that these two were once bacteria. They have their own double membrane, just like the cell membrane or plasma membrane. They have their own DNA, and they're actually able to replicate their own DNA and make proteins as well. One other thing that we want to notice here is that scientists do believe that the first eukaryotic cell, scientists do believe that our mitochondria did come first, because all eukaryotic cells have mitochondria, and not all of them have chloroplasts. But one thing we also want to keep in mind here is that as the cells became more advanced, as they continued to change over time and evolve, they did end up getting many different compartments or divisions for their different cellular activities. So when we start to look more at cell organelles, we'll see that all these different compartments allow them to be more efficient and allow them to be more specialized as well. So if we'll continue on to the next portion, we're actually going to look at how do those membrane-based organelles aid in the functioning of these cells. What we're actually going to look at today is the endomembrane system. So the endomembrane system is going to be a network of organelles that participate in synthesizing, folding, and transporting of proteins. When we look at the endomembrane system, we're actually going to be looking at the nucleus, the ER, and the Golgi. At least in this section, an endomembrane system could include any of the internal membranes within the cell, and also including a little bit of the plasma membrane at the very end. But these are the structures that we're going to focus on today. What you want to know is that the ER is actually a continuation of the nucleus. It's connected to the nucleus. Endoplasmic actually means within the cytoplasm of the cell, and reticulum is a network, okay, or a net. And so all of these, to be able to identify, you can see little connections between those folds of the ER. Now we have, first of all, the nucleus. And so the nucleus is going to represent, or is going to contain, our genetic code. And within the nucleolus, we'll actually make our mRNA. And mRNA is the carrier of the protein code, or of the amino acid sequence code, okay? So we're going to start with our nucleus first. From the nucleus, and we're talking about how these interact with each other. From the nucleus, next we're actually going to have our rough ER. Now our rough ER has little dots on it, these little dots here, and those represent ribosomes. So ribosomes, some ribosomes, are actually attached to the rough ER. They're not static, they can actually come off and on, but they are there and they are attached. Because of the location of this makes them really, really close to that genetic material, and that is an advantage. But with the rough ER is actually able to make and modify or fold proteins because those ribosomes are right there. The ribosomes will actually produce that amino acid chain. The amino acid chain would go within the rough ER, within the network, and the folding would actually occur within it. The rough ER can actually also produce the secretory proteins. And so this actually means that the rough ER a lot of times is involved in making proteins that will leave the cell and not stay within the cell, like the free-floating ribosomes would. Free-floating ribosomes often make proteins that are going to stay within our cell. And then our rough ER will also make glycoproteins, or proteins that have specific tags on them. Those tags will identify them and allow them to be transported to specific locations. In connection with the rough ER, you have your smooth ER, okay? So your smooth ER would be kind of the third step in this process. The smooth ER is actually involved in several things. It is involved in lipid synthesis. So it can actually make some phospholipids as well. Because it can make those phospholipids, it can actually make vesicles. So little transport compartments, okay? If you notice right here, here's our smooth ER, here's a little vesicle coming off of it to transport the proteins. And it can also, the smooth ER can also be involved in detoxifying compounds that enter our cell. So alcohol and actually some drugs as well can be broken down by the smooth ER. The smooth ER will actually add a hydroxyl group to certain drugs, making it more polar, making it more soluble, so it's more likely to flush out of your system. But from there, you'll actually have your Golgi. The Golgi technically will be the fourth step in this process. There's two sections to the Golgi, the cis and the trans. Cis just means that it's going to be closer to the nucleus. And then trans, the fifth portion, will be far from the nucleus. Okay? So that's just showing the direction. What it's really showing you is that the cis end of the Golgi is going to be receiving some of those vesicles or some of those proteins from the smooth or rough ER. And then within the Golgi, we'll actually have some modification of the proteins, and it'll also ship out the proteins. It can also store them for a little while as well. But once they modify and get them ready to be shipped out, they'll actually be shipped out from the trans end of the Golgi. And sometimes those transport vesicles can move towards the end or the lining of the cell membrane, the plasma membrane, and diffuse out of the cell that way or leave the cell that way. And so often when you have proteins that are leaving the cell, these are proteins that can help the organism as a whole, and they're not just working on something within the cell. Okay? And so remember, for this section, you really want to think about how these membranes are working together, how they're interacting with each other to do this one big job, which is to make the proteins and to ship them out to where they need